Dear students, now I am going to start the chapter Translation. Today I am going to discuss about genetic code. You have studied already the process of the molecular biology, the concept of molecular biology, central dogma. Replication, transcription and translation are all the three main processes used by cells to maintain their genetic information and to convert genetic information into gene products. They may be proteins or RNA. So you know that is the three main key concepts of molecular biology. DNA acts as a template for its own synthesis, that is called replication. So it is a template for the production of RNA synthesis. RNA translated at the level of ribosome into the amino acid sequence of the proteins. So all this in this cartoon also the same thing you can see. DNA transcription, mRNA, and get translated at the level of the ribosomes into the proteins. So genetic information on DNA as a series of genes. Gene expression is how cell decodes these genetic information to synthesize proteins required for cellular function. Hence, gene expression involves transcription and translation process. RNA and proteins synthesized in organisms during development process or else during the maintenance or else in response to the environment. So the RNA or transcription and translation occurs and they are responsible for organisms' characters. So what is genetic code? So translation is the process of translating the sequence of mRNA into the sequence of amino acids during protein synthesis. Those sequence of mRNA is get translated into the sequence of the amino acids of the proteins. So genetic code describes the relationship between the sequence of base pairs of a gene and the corresponding amino acid sequence that it encodes. So it is a relationship between the sequence of the DNA or the gene that of the protein synthesized. So in the cell or cytoplasm, the ribosome reads the sequence of mRNA in the triplets, in the form of triplets or three bases to assemble the proteins. So in the mRNA will be read in terms of the the three lettered or triplet codons. So and each triplet codons specify in amino acid. So this you will able to see in this cartoon. So the instructions in a gene is in the form of A, C, G, T nucleotides, that is on the DNA. So genetic code describes is the relationship between the nucleotide sequence of mRNA and the sequence of the amino acids in the polypeptide chain. So group of three bases of mRNA specifying one amino acid is called triplet codon. Genetic information of DNA is translated into 20-lettered code of amino acids which are the building blocks of proteins. So all biological proteins, if you analyze, they are made out of 20 types of amino acids. So these genetic information on DNA, they are get translated into the 20-lettered code of amino acids which are the building blocks of proteins. So genetic code is a set of 3-letter combinations of nucleotides called codons, each of which corresponds to a specific amino acids. So for example, AUG codes for methionine, UUU codes for phenylalanine. So in this way, this is the 3-lettered code which specify a amino acid, and or else there are 3 codons, they specify the stop codons. Triplet codon concept, this is actually proposed in way back by the Francis Crick and his group. Later, the genetic code, the term coined by G. Gamow. So in 1961, it is a very interesting experiment. You should go through the details of these experiments. It's a wonderful experiments of Nirenberg, Mathai and Haragobinda Khorana. So they deciphered the genetic code and described the triplet codon and its corresponding amino acids through in vitro protein synthesis in experimental setup using artificial homo and heteropolymers of mRNA. So Nirenberg and his team, they used homopolymers and like UU like that, you can see they could able to synthesize only phenylalanine. Then Haragobinda Khorana, so Indian origin scientist, so he worked on heteropolymer and he able to decipher the the codons for all the amino acids. So all possible codons also. So that's why for their great work, so Haragobinda Khorana and Nirenberg and Robert Hooley together, they were awarded Nobel Prize in 1968. So this is what the interception of genetic code and its function. So that is for that they are awarded with the Nobel Prize. So the Haragobinda Khorana, he studied in Punjab University and later he moved to the US for his research, and he is the third person in India to get Nobel Prize. He awarded Nobel Prize in 1968. So US government also they have in the memory of Haragobinda Khorana, he died in 2011, and they have a Khorana scholarship for Indian students, summer scholarship for Indian students. So genetic information of DNA is copied in the form of mRNA by transcription. Then this message of mRNA expressed in the form of a protein during the process of translation. You know all this, the process we have discussed in during transcription. So genetic code refers to the system of coding of amino acids as a group of 3 bases or codons of DNA or RNA. So this is a method of coding of amino acids. Since there 20 different amino acids in protein which are specified by 3-lettered combination of ATCG bases. So that results by permutation and combination 64 possible 3-lettered nucleotide sequences, and these 64 codons, so 61 represent the 20 amino acids and 3 are stop codons. So during the translation, the universal genetic code of triplet base sequence is used which specify a kind of amino acid. So this is what you can see, the genetic code. So here the triplet code UUU for phenylalanine, UCU for serine. So likewise you can see the this genetic code, and this genetic code is almost nearly it is universal. The same code is used even in the E. coli. For the amino acid is the same code even in the human or else in the any flowering plant or any plant, you know. So this is what you can see, the triplet codon, 61 codons, they are specifying 20 amino acids. You can see some amino acids like leucine, they are coded by or specified by more than one triplet codon. So similarly you can see the threonine, serine, proline. So only you will find the methionine specified by only one codon, and similarly the tryptophan is also, this is the one, it is a specified it's a tyrosine. Tryptophan is also specified by only one, this is this one. So this is what you can see, the genetic code, how it acts exactly. Then what are the properties of genetic code? So genetic code is triplet in nature. group of 3 bases of mRNA specifying one amino acid is called the triplet codon. You will see here. So, AUA isoleucine, CGA arginine, GUC valine likewise. So, each of the 20 amino acids in the protein is specified by one or more nucleotide triplet codons in mRNA. Out of 64 codons, 61 specify amino acids and 3 signal for chain termination or stop codons. So, those stop codons, you know that, so they are the UGA, UAA, UAG. These are stop codons or terminator codons. So, code is unambiguous. So, they are very specific to a particular amino acid. So, that is what unambiguous. Each triplet specifies an amino acid. So, non-overlapping, code is non-overlapping. The adjacent codons do not overlap. The genetic code is read in words of 3 nucleotides. After reading one triplet, the reading frame shifts to the another. So, AUA, so it codes for isoleucine. Next it shifts for coding CGA, it's for arginine. Next reading frame shifts to GUC, that is for valine. So, it is from 5 prime to 3 prime. So, this genetic code will be read by continuously GAC, UCA, CUG, ACU, like that you can see. So, this is what you can see the meaning overlap, non-overlapping. So, the genetic code is comma free or commaless. It has to be read continuously starting from 5 prime to the running to the 3 prime. The genetic code read continuously without any break. So, during protein synthesis they will be read continuously. Genetic code is you can see read in a 5 prime to 3 prime direction during transcription and as well as in the translation process, you can see. So, this is what you can see, DNA during transcription, then during translation you can see AUG methionine, CCG proline, then you can see the AAU, CAA, so glycine, likewise you can see it will be read continuously. So, the important property of the genetic code, the degeneracy of the genetic code or redundancy it is called. Genetic code is described as degenerate or redundant because a single amino acid may be coded more than one codon. Out of 64 codons, 61 codons represent 20 amino acids. So, several codons specify same amino acid. So, for example, leucine, serine, arginine, they are specified by 6 triplet codons, whereas proline, valine, threonine, glycine, alanine, so they are specified by 4 triplet codon. Each will specify by 4 triplet codon. This phenomena is called redundancy of the, so they are called the synonyms, you can see. Some people will similar how exactly they have their actually they have one original name and they are called by several other names. The person is the same. Similarly you will find they have, so there is a you can see the synonyms, the codons are synonyms, you can see. This phenomena is called redundancy of the code or degeneracy of the code. Except methionine and tryptophan, they are specified by only one amino triplet codon. Methionine specified by the AUG. So, and rest of this, all these are you can see they have specified by more than one triplet codon. So, methionine by AUG and tryptophan by UGG. So, the genetic code is degenerate. What is the advantage of this? Mainly in the 3rd position, you will find able to see here glycine. It is the first and second the bases are same, GGG. The only variations you will find in the 3rd base. So, what is the significance? Usually mutation occurs in the single nucleotide in the 3rd position usually. So, that will be not leading to any structural changes or functional changes. If for example, so this GGG, if it is the third nucleotide is replaced by A, then it codes for the glycine only, it is not coding for other amino acids. So, there is no structural or functional changes that occurs in the proteins. So, when in so the resulting polypeptide will not structurally and functionally different from its kind. So, that is the advantage. So, otherwise what happens, you can see it will be very difficult to control the mutations. So, here the genetic code, the structural itself, it is it controls the mutations because of the same amino acids are specified by more than one triplet codon. So, the genetic code is universal. The famous sayings of Jacques Monod, what is true for E. coli, it is true for the elephant. That means genetic code, whatever the codon for amino acids in E. coli, the same will be in the elephant. So, in all organisms, genetic code is the same. Each codon specify a single amino acid. However, there are exceptions like UGA, a stop codon actually, it is coding for selenocysteine and in cases, and tryptophan in case of mycoplasm. So, similarly AUA, isoleucine, it is coding for, but it codes for methionine in mitochondrial DNA. So, AUG and GUG encode for methionine, you can formyl methionine if they are in the beginning, acts as a start codon, but they may encode methionine or valine when they are present in the middle of the mRNA. So, this is what you can see, the universality of the genetic code. So, it will be the same for all organisms with few exception, you will see. So, genetic code is the strongest evidence that all living organisms have a common evolutionary stem. From that you can see they have branched out. So, genetic code contains start and stop codons. The codons which initiate protein synthesis called start codons, that is the AUG. The first amino acid polypeptide chain is always synthesized is methionine. It is specified by the triplet codon AUG, is the start codon. Then the prokaryotes AUG codes for a modified amino acid, it is called formyl methionine or FMET. So, the protein synthesis stops by the UAA or UAG or UGA. This is the stop codon you will find at the end of the mRNA at the 3' region. When this codon arrives at the acceptor region of the ribosome, then the protein synthesis is terminated. So, it is as it is not specifies any amino acids. So, this is about the genetic code and the properties of genetic code. So, this is how the Har Gobind Khorana is played a major role in deciphering this genetic code. So, this has made a basement for the development of the entire the bioinformatics. Thank you.